Hello, science students. This is Dr. B, and we're doing deer mouse fur color from the field to the beach, activity 13, lesson 5.1. We're on the home stretch here. We're doing population genetics today. Working with your understanding of the effects of the MC1R alleles. Those are the alleles for making the receptor gene in the protein that either grabs onto the signal protein and breaks it in half or doesn't grab on at all, and either signals to make, in the dark version, it signals the cell to make dark melanin, the eumelanin, and in the light version, it signals the cell only to make pheomelanin. You'll learn about tracking and measuring evolution using relative frequencies of alleles in a population. This time we're going to look at alleles, not just mice in general, because now we know different kinds of mice have different alleles, and so we're interested at the allele level. We're looking more at genetics this time. So in Unit 1, you examine data from a scientific study on different mice in different populations in the southern United States. You also used a simulation to graph how populations of mice change over time with and without predators. Remember the hawks? When you recorded the number of mice in your data tables, you were recording the frequency of the mice with each fur color. You also calculated the number of mice, that fur color, relative to the total number of mice in the population by using this formula. The number of mice with light fur, total number of mice in the population. You then multiplied the result by 100 to get to percentage. This process is also known as finding the relative frequency. Relative frequency can be written as a fraction, decimal, or percent. The relative frequencies of fur colors can change in a population over time, especially when predators act on that population, or the environment can also act on that population, or other factors that select for certain alleles. This is a set of simulated photos of a population of mice over time. So they live on the beach. It's a light environment, has a light background. Eventually, the mice, through the action of predators, probably matched their background, but it probably didn't start out that way. Which group of mice is the darkest? And when we looked at this as a class, we decided B was the darkest and D was the next darkest. And then we decided A was kind of in the middle, which probably led to C, that led to E. You can come up with your own order. It's just important that you justify your decision making. For question number 2, we explained our reasoning in class. We started with the darkest mice, assumed there was a mutation that led to lighter fur, and that the hawks acted to select against, or basically they ate the dark mice before they could reproduce, and there were more light mice that survived, and over time, as that process occurred, more and more light mice became part of the population. In reality, there are far too many mice in a population to count and analyze each one. Therefore, scientists have strategically collect and study a smaller number of mice or a subset from the population and use calculations to estimate the traits of a full population. And this way, they estimate the relative frequency of the traits in a population. Now, let's put that in a way that's maybe relevant to students. Let's say the population of mice might be your whole neighborhood, several blocks. You don't want to go out and collect all of the mice for 10 blocks in your neighborhood. You maybe just want to collect mice for 4 or 6 lots. Lots is like a little plot of land where a house is. And collect a subset of those mice that live in your neighborhood, or a smaller group instead of every mice that lives in the 10-block neighborhood. And then you can look at the relative frequency. You can figure out what percent dark mice, what percent light mice, if you have a dark and light mouse population in your neighborhood. A population has changed over time. Below are samples taken from 3 different points in time. Here's time 1, time 2, and time 3. For each point in time, count the number of mice with each fur type and calculate the relative frequencies of the fur colors. We're going to record our results in lesson 5.1, table 1 of your field notebook. So here's your field notebook. Yours might be in Google Docs. This one happens to be in Word. So lesson 5.1, and we have a reminder for relative frequency. We can do it as a fraction or a decimal, putting the number of specific color of mice, like dark, light, medium, over the total number of mice. Or we can do that and multiply times 100 to get a percent. So when we look at time 1, we want to count the total number of mice. So we count 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's 9 total mice. And then if we look at the next here, we're supposed to count the number of light brown mice. And I see 3, or light, 1, 2, 3. So I see 3 light brown mice. And then I calculate, there's 3 divided by 9 times 100 equals 33.3%. So I took the number of light brown mice and I divided by the total number of mice times 100 to get 33%. I did the same as you go down to medium brown mice. I counted 1, 2, 3 out of 9 total mice. So 3 out of 9, 33%. Dark brown mice, there just happen to be the same number of dark brown mice here. So 3 out of 9, 33%. Time 2 was a little bit different. This time, light brown mice were 1, 2, 3, 4, 5. 5 out of 10 is 50%. So that's the number of light brown mice divided by the total number of mice. So 5 divided by 10 times 100%. For medium brown mice here, I noticed there were 3 out of 10. 3 divided by 10 times 100 is 30%. And the dark mice are 2. 2 out of 10 times 100 is 20. And you can do time 3. So that's our first table. Lesson 5.1, Table 1, Calculating Relative Frequency of Fur Colors Over Time. Warmed up, I gave you a hint to explain in your own words to how we calculate the relative frequency of a fur color in a population. Here's the basic formula, and you can write it out however you prefer. Let's go to the next page. So we're here on page 2. Now, instead of just mice, we're going to look at the frequency of alleles. And unit 4 that you found out a mouse's fur color is determined by alleles. That's versions of a gene. Everybody gets the same fur color gene, but they get different versions. Some mice get RD, which is the dark version. Some get R light, which is the non-functional version. It doesn't break open those signal proteins, and it doesn't tell the cell to produce that dark pigment. The mouse inherits the alleles from the parents. These alleles code for proteins that are involved in the production of eumelanin, that's the dark pigment, and pheomelanin, that's the light pigment. You also use the alleles to describe a population. To measure the changes that occur in a population over many generations, scientists collect data on the genotypes of a sample of individuals in the population, like you going out and grabbing all of the mice maybe from 4 or 5 lots in a neighborhood instead of 10 blocks. Then they calculate the relative frequency of alleles for a particular gene over many generations. The change in relative frequencies of alleles is a way to measure the genetic change in a population. So we're looking at how the environment, whatever factor that's selecting some mice and selecting against other mice, like hawks eating a mouse is selecting against it because it doesn't survive to reproduce. So let's go over this example here. Consider, for example, how to calculate the relative frequency of alleles in a mouse population. So we have 3 mice in this population, 2 dark mice who both have copies of the dark allele, and one medium mouse who has a copy of dark and a copy of light. Because each mouse has 2 alleles, we can count 1, 2, 3, 4, 5, 6, or we can just go 3 times 2. 3 mice times 2 alleles each equals 6 total alleles. We can count the dark alleles, 1, 2, 3, 4, 5. If we want to know the frequency or the relative frequency of dark alleles, we take 5 dark alleles over the total number of alleles, that's 6, is equal to 0.83 or 83% when you multiply times 100. The number of light alleles gives us a relative frequency of 1 divided by 6, which is the total number of alleles, and that gives us 17%. You'll notice that 83 plus 17 equals 100% because we only have 2 options here, dark and light, and they need to add to 100%. In our own words, explain how to determine the relative frequency of alleles. Now, I've capitalized the word alleles here to help you remember that this time we're not counting mice, we're counting alleles. So each mouse has 2 alleles, and sometimes they're different in the case of medium mouse, and sometimes they're the same. In the case of dark mice, they have 2 dark alleles, and light mice have 2 light alleles. So we count the number of total alleles in the population, or in our sample, which is 2 times the number of mice because each mouse has 2 alleles. Then we count the number of light or dark alleles and divide the light or dark alleles by the total and multiply by 100 to get percent. You will now calculate the relative frequency of alleles in a population over time. You may notice that you can use the fact that every mouse has 2 alleles as a shortcut to help you. We're going to record our results in lesson 5.1, table 2 in our field notebook. So let's go back to our field notebook and do some calculations. So this time our formula is a little bit different. Remember, we're dealing with alleles instead of mice. So we have to make sure that we're counting the alleles, not just the physical bodies of mice. The relative frequency as a percent is just the specific allele divided by the total alleles in the population divided by or multiplied by 100. This time we're working in table 2. And let's just count the total number of alleles in each population. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 mice times 2 alleles each equals 18. We can do the same for time 2. There are 10 mice times 2 alleles equals 20 total alleles in this sample. And time 3, we've got 8 mice, so times 2, that's 16 alleles. So now let's look at our light. So to figure out the number of light alleles, let's find the light mice first. So we know each light mouse has 2 alleles. So that's 1, 2, 3, 4, 5, 6. So just to keep track of the number of alleles and make it easier, let's put a 2 on every light mouse to remind us there's 2 light alleles in the light mouse. And then we know dark mice have 1 of each. They have 1 dark and 1 light. So if we're just counting light here, we count 1 here in the medium mouse, 1 here in this medium mouse, and 1 here. So if we add these all up, we get 6 plus 3 is 9. So we get a total of 9 light alleles here. We take the number of light alleles, that's the light alleles, and we divide them by the total times 100% equals 50%. So half our alleles in the time 1 population are light alleles. Let's do the same for time 2. Let's figure out how many light alleles are in time 2. We've got 2 in every light mouse. So since we have 5 light mouse, that's going to be 10 light alleles. And then we have 2 medium mice, and we know each medium mouse has 1 dark and 1 light. So that's 1 light allele for each medium mouse. Now if we add up everything here, we have 1, 2, 3, 4, 5 twos, that's 10, 11, 12. Oh, 13. So we have 13 total light alleles here. We have 9 here. We're counting alleles, remember. And then when we go over here, the population gets lighter, so we have 2 alleles for every one of these 6 light mice. So that's 12 light alleles so far. And then we have 1 more here, so that's 13, and 1 more here in this medium mouse, so that's 14. So there's 14 light alleles. We divide that by 16 total alleles. 14 divided by 16 times 100 equals 87.5%. I'll let you do the dark alleles. I'll give you a hint on the first one. We'll count dark alleles. Let me erase all the light allele numbers. I'll choose a different color. For dark alleles, we know that every dark mouse has 2 dark alleles. So there's 3 dark mice. That's 6 dark alleles so far. We have 1 in every light mouse. We have 1, or in every medium mouse, we have 1 dark allele. Remember, medium mice, I have 1 light and 1 dark. So we just count 1 if we're counting dark alleles. So we have 3 twos, that's 6, 7, 8, 9. We have 9 dark alleles here. I'll get you started on this one. And you can do the last one. Now it's our mission to fill out some pie charts for each of these populations. So I'll do the first one. Population 1, we want to label our graph. Allele frequency. And you might have to stagger your titles here a little bit. And so in population 1, we know we had 50% light alleles. So we color in 50%, or 9, I believe it was 9 out of 18. Yep, 9 out of 18. You'll notice that this pie graph is divided up into 18 sections. So we just color 9 of them for the light mice. And we'll make light mice yellow here. So I'm not going to color it all in. You can do a better job if you have a graphics program. So this part of the pie chart, all of this is going to be light mice. And we know since there are only 2 alleles, we can color the other half as dark mice. And if we put a key, we have a key down here at the bottom, and we should put our colors in here and the key. So we're using this color for dark mice, and we're using yellow for light mice. So you can do something similar on your own paper. I'll let you do the other 2 pie charts. Notice this one is divided up into 10 and 10 sections, or 20 total sections for the 20 alleles in population 2. And population 3 here is divided up into the number of sections that you have, the number of alleles. Let's the third graph here, the time 3 population graph, is divided into 16 segments, and so you can color in the right number of segments for light and dark for the population 3 graph. Make sure to label all of your populations. So make sure you have population 1 for here, population 2, and population 3. So label your graphs, make sure you fill in the key with what colors you use to fill in those pie graphs as well. So, we have an investigation. A scientist is studying the mice in 4 different populations located in Florida. The scientist has calculated the frequency of the R light allele in each of the 4 populations and recorded them below. A sample of the mice is taken from one of these 4 populations. Examine the sample closely. Okay, so we have 1, 2, 3, 4, 5 dark mice. We have 4 medium mice, and 1 light. From what population was the sample most likely taken? Defend your answer. Well, let's figure out how many light alleles are in this population. We know there are 10 mice, so there are 20 alleles. So let's figure out the relative frequency of light alleles. So we can, if we know the number of light alleles, we can divide it by the total and figure out relative frequency of light alleles and try to match it to one of the scientist populations. So let's grab a color here. We'll do light alleles in pink since it's easy to see. We know this light mouse has 2 light alleles, and every medium mouse has 1. Because remember, medium mice have 1 dark allele and 1 light allele. So if we add up the total number of alleles here, we have 2, 3, 4, 5, 6. So we have 6 light alleles over 20 total alleles, because there's 10 mice times 2 is 20. And then that's T for total. And then if we multiply that times 100, I believe we'll get 30%. So if we got 30%, which one is that closest to? Is it closest to 15%, 32%, 40%, or 53%? So in your answer, you should explain this calculation, and then you should explain why you think it's closest to whichever population you've chosen from this group here. So for number 5, explain the calculation, explain how you got the number of alleles, and pick which population you think it's closest to. This is the end of page 2. And we'll continue on page 3 in the next video. We're going to do our own investigation next time. I'm pretty excited. I'll see you in lesson 5.1, part 2.